Welcome to Part 3 of the Cell Cycle Regulation series. Today we study checkpoint machinery controlled by tumor suppressors such as p53 and Rb, the genome's defense system that prevents cancer. Checkpoints act like exam gates. A cell must prove it is safe to divide. They stop replication of damaged DNA, ensure correct chromosome alignment, and either allow repair or trigger death if damage is severe. Without these gates, cancer can arise. Memorize this table. Rb directly controls G1S by restraining E2F. ATR-CHK1 respond to replication stress, ATM-CHK2 respond to double-strand breaks. MAD and BUB proteins sense unattached kinetochores and block APCC activation via CDC20, preventing premature sister chromatid separation. Now let's look at one of the most fundamental control gates of the cell cycle, the G restriction point, controlled by the Rb-E2F pathway. In early G, the protein Rb, or retinoblastoma protein, tightly binds the transcription factor E2F. When Rb is attached to E2F, it acts like a brake, preventing the cell from expressing the genes required for S-phase entry. So the cell cannot replicate DNA until this brake is released. As growth signals arrive, the first cyclin CDK complex to respond is cyclin D, CDK4/6. This complex begins hypophosphorylating Rb, meaning it adds a few phosphate groups. This partially weakens Rb's grip on E2F, but doesn't fully release it yet. A little later in G, cyclin E, CDK2 becomes active. This complex now hyperphosphorylates Rb, adding multiple phosphates. At this stage, Rb becomes fully inactivated and releases E2F completely. Once freed, E2F acts as a transcription activator, turning on all the genes needed for S-phase entry, including those involved in DNA replication machinery. This moment, when Rb releases E2F, is the famous R point, or restriction point. After crossing this point, the cell is fully committed to DNA replication, even if growth signals disappear. And that's why the Rb-E2F checkpoint is so critical. It ensures the cell only enters S-phase when conditions are safe and the environment is favorable. Now let's decode one of the most important pathways in cell cycle regulation, the DNA damage checkpoint, controlled by the guardian of the genome, p53. Everything starts with DNA damage. If the cell has a double-strand break, the first responder is ATM. If the cell has single-strand breaks or replication stress, the responder is ATR. These two sensors are recruited to the damage site with the help of the mRN complex. Once activated, ATM triggers CHK2, and ATR activates CHK1. These checkpoint kinases now phosphorylate p53. This phosphorylation is extremely important because it protects p53 from degradation by MDM2, allowing it to accumulate. Stabilized p53 now acts as a powerful transcription factor. One of the first genes it induces is p21, a strong CDK inhibitor. And once p21 rises, it halts cyclin CDK activity, putting the brakes on the cell cycle either at G1S or G2M, depending on when the damage occurs. At this point, the cell evaluates. Can we repair the DNA? If repair is possible, p53 keeps the cell arrested while repair enzymes fix the damage. Once corrected, the cell cycle resumes safely. But if the damage is too severe and beyond repair, p53 pushes the cell into its second role, activating BAX and BAK, the mitochondrial apoptosis pathway. This ensures that severely damaged cells do not divide and cause mutations or cancer. So in summary, ATM and ATR sense the damage activate CHK kinases stabilize p53 induces p21 CDKs are shut down cell cycle arrest and repair. If repair fails, p53 commits the cell to apoptosis. This is why p53 is rightly called the guardian of the genome. Now let's explore the spindle checkpoint, also called the mitotic checkpoint, the last and most crucial safety barrier before a cell enters anaphase. At this stage, the cell is checking only one thing. Are all sister chromatids correctly attached to spindle fibers at their kinetochores? If even one kinetochore is unattached, it immediately recruits the checkpoint proteins MAD2 and BUB-R1. These proteins bind to CDC20, forming an inhibitory complex. And this blocked CDC20 means the major mitotic regulator APCC remains inactive. So the cell is essentially holding its breath, preventing anaphase from starting. But once every chromosome is perfectly attached and under tension, the inhibition is released. CDC20 is freed, and APCC becomes fully active. Active APCC now triggers ubiquitination of securin. Securin is degraded by the proteasome, and its destruction unleashes the enzyme separase. Separase cuts the cohesin rings, allowing sister chromatids to separate and move toward opposite poles. At the same time, APCC also targets cyclin B for degradation. Cyclin B destruction inactivates CDK1, allowing the cell to exit mitosis smoothly. So in summary, the spindle checkpoint acts like the final security gate. It ensures perfect chromosome attachment, prevents aneuploidy, and guarantees accurate segregation before the cell completes division. Checkpoints decide cell fate. Small damage is repaired, then the cell resumes. Severe, unrepairable damage leads P53 to activate pro-apoptotic factors such as BAX, triggering programmed cell death and preventing propagation of mutations. Checkpoints decide cell fate. Small damage is repaired, then the cell resumes. Severe, unrepairable damage leads P53 to activate pro-apoptotic factors such as BAX, triggering programmed cell death and preventing propagation of mutations. Remember these keyword pairs: ATM for DSBs, ATR for replication stress, CHK kinases for signaling, P21 as the CDK brake, and APCC as the anaphase trigger. These are CSIR-NET favorites. Checkpoint biology is central to cancer therapy. CDK4/6 inhibitors like palbociclib, ribociclib slow cell cycle progression in hormone receptor-positive breast cancer. Aurora kinase inhibitors and PARP inhibitors exploit mitotic and DNA repair vulnerabilities. P53-based therapies remain an active area of research. Thank you so much for watching. If you're enjoying this cell cycle regulation series, don't miss Part 4, where we dive into DNA damage response and repair pathways. It's one of the most powerful topics for CSIR-NET, GATE, and all advanced biology exams. Make sure to subscribe so you don't miss the next class. Smarter biology learning continues right here on Sravika's Biology.